The next experiment tries to give an indication of lipid content in different sources of lipids. Now, when we want to extract lipids from a food source, for instance, like nuts, butter, or milk, you cannot extract it using water because lipids are water insoluble, and so we use ether, which is a solvent that is hydrophobic, in order to extract these substances. Because everything is in ether, I cannot use a pen or a marker to label anything because the ether will dissolve the ink, and so I'm using a pencil, and with a pencil I drew 5 circles on a filter paper. Now I'm going to label them with the different food extracts that we have. We have flour, we have
butter,
butter,
yeah,
milk, and nuts, castarat. Our control cannot be water. I explained why. So our control is ether, okay, which is the solvent that we use to extract the material. So now I'm going to get these extracts. The extracts are kept under the hood because of the heater and the ether fumes, and so we're going to put the hood on, and we're going to get a drop of these extracts for our filter. I have flour extract. So this flour has been mixed with ether in order to take the fats from the flour and dissolve them in ether. We have milk, nuts, and our control ether itself. Okay? So here we go. Flour. Oh no.
It's okay. It's still in the same location. So you need
Butter. This time I don't spill it everywhere, I spill it. Because the ether doesn't.
yeah, I suppose the way it's okay.
Milk. Let's bring this closer.
So you need to tilt it maybe.
Oh.
You need to tilt the paper. Yeah. Because it spills.
Nuts. Doesn't matter, we're going to put the Sudan green, the Sudan red coffee. You see, it doesn't
Yeah.
Nuts. So. And ether. Okay, now we let it dry a little bit. Ether evaporates very quickly, so it dries very quickly. As you can see, my dropping wasn't very precise. The ether runs out from the pipette, but it doesn't matter because we're going to be more precise with the Sudan red. Now, in order to identify the fats, I put Sudan 3 like we did before. I'm going to add a few drops of Sudan 3 to the circles. So we have here the flour, two drops is enough, butter, milk, nuts, and ether. Butter didn't mix very well. Let's mix it. Okay. Now we need to wait a bit to let this dry. Yeah?
Okay.
In order to be able to identify different intensity in the red color. Sudan 3 binds with the fat, and it will give a different intensity of redness according to how much fat is present in the substance. Now, remember what is our control here. Our control is ether, okay? So when we want to compare the intensity of the color, we want to compare with the ether spot, which is our negative control, okay? There is no fat in ether, there is just ether, okay? So here is our negative control. We wait a little bit for this to dry. As you can see, flour can be compared to ether. It might have a slightly darker color, but you know, flour basically has no fats in it. Butter, on the other hand, you can see here strong orange color. That is indication of presence of fat in butter. Milk has a little bit more than ether. Between ether and butter, yeah, so there is some fat in milk. And nuts also have a relatively large amount of fat, almost comparable to the one of butter. As you can see, compared to the negative control, much more fat in nuts and butter compared to flour and milk. If I were to write these experiments, the result of these experiments, you will do it on a notepad, but if I were to write it on this filter paper, I would write a minus for the flour as negative, because there is no here. I would write 3 pluses for butter. As you can see, the intensity of the color here is quite dark. I would write 1 plus for milk, and 2 to 3 pluses for nuts. Yeah, this would be how I summarize the results of this experiment. Remember, this is my negative control.